Hi, welcome to Chapter 9. Now that we are familiar with what we described as normal cell division, known as mitosis, let's move to have a look of another kind of cell division process. And this type of cell division is of course meiosis. We use meiosis to produce sex cells. So these are our sperm cells and our egg cells, the very two cells that come together and then generate a new individual. And there are some special characteristics that we need to have for sex cells. Let's look at those. First of all, people look very different from each other. We all are unique individuals. With the exception of identical twins, we all not only look different, but also have different genetic composition. Our DNA is just as unique as our fingerprints are. Remember, these were those bases of nucleic acid molecules that formed these horizontal bars of what looked like a stepladder twisted around itself. And really, it is the order of those bases that code the uniqueness of each individual. So the first thing that meiosis needs to provide for us is making sure that each time a new individual is made for the very first time by sperm and egg cell fusing together, the genetic composition of that individual is going to be something brand new. I often think of it as taking part in a huge genetic lottery. We try to hit this genetic jackpot of making a brand new individual which has a combination of genetic material that makes it more successful than any other individual that has existed so far. Doesn't this sound a little like evolution? Well, to do this, to make a genetically unique new individual, we get this genetic material from our mom and dad. So you get an egg cell and a sperm cell. And this has some special consequences. Let's consider those, but first go back to this slide that we have seen before. We remember that each normal cell has typically a full amount of genetic material. This is what we know as the cell being diploid. It has 100%, or what we know as 2N, of all the genetic content. This is 23 pairs of chromosomes. But if this is the case in a normal cells, these ones that we discussed by mitosis, the process we talked about in our last module, there must be something different in making a new individual by combining two cells together, the sperm cell and egg cell. So when sperm cell and egg cell join together, both of them carry only 50% of the final genetic material of a new individual. Otherwise, the cell's nuclei would get more and more cramped with DNA and all sorts of genetic problems would arise. So each sex cell carries half of the genetic material, and now they are known as being haploid, symbolized by letter N. You will notice that just as 50 plus 50% makes 100%, N plus another N makes 2N. So this is the first challenge that the process of meiosis must meet. We must halve the amount of chromosomes in a sex cell during this process of making this. The second challenge that we must meet, I have already kind of referred to. We need to produce unique sex cells so that we would end up with all these unique brand new combinations of genetic material which give rise to a brand new unique individuals. Remember, we were trying to win that genetic lottery of being an individual that has never existed before and that is able to survive in the environment better than any other individual has done before. It is indeed what evolution is all about. This is why I think that biology is so much fun. Really, all these concepts that we all talk about relate to each other. We would not be talking about this in chapter 9 if we had not talked about evolution in chapter 1. So we have already discussed about how the process of meiosis has to meet these two special characteristics of this kind of cell division. Remember, mitosis, our normal cell division, doesn't have to do this. So we'll find out in this chapter that meiosis will be a little bit more complicated than mitosis was. In this chapter, we will study the mechanisms that allow us to meet these two goals. It'll be fun. So, to start our story of how do we do this, let's go back to this slide from our previous chapters. We have already talked about how DNA is packed into chromosomes inside the nucleus of a cell, right? Well, it turns out that each human individual has 46 chromosomes, or as we often prefer to say, 23 pairs of chromosomes. And this picture that you see here is what we call a karyotype. It is an important tool that geneticists use. Basically, it just lists all the chromosomes of one cell in an order as arranged by their size. And we can see that each of these contains 2 chromosomes. Remember, a chromosome was this X-shaped structure. So we have 2 of these for each one. And these 2, most of the time, always look fairly similar, don't they? They even have areas that appear similar in these pairs of chromosomes, as highlighted by the pink top region of chromosome pair 5 that we have enlarged here. So both chromosomes are made of chromatids, and all chromatids have pink top regions in this example. We call these chromosomes as being homologous. This means that they are same, both in terms of their structural appearance, but also in terms of the type of information that they carry. So same size, same shape, same location, and so on. But although you find the same locations in all chromosome pairs, and both chromatids of a single chromosome, they must not carry the same instruction for the output protein or trait. This might sound a little complicated, but let's look at our example here. So you remember that we noted that for chromosome pair 5, they all had pink top parts at the very end, right? And that this is not really the case, but for the talking's sakes, let's imagine that these pink regions would be the site where we carry instructions for whether you get freckles or not. And although they might look similar, one of these chromatids may have instruction recorded in it that, no, you will not make freckles for this individual, while the other one has a set of instructions that, yes, you will make freckles. In this case, this set of instructions for whether you get freckles or not would be 2 different forms of alleles that you get from that gene. If this sounds complicated, do not worry. I have a nice video coming up explaining this next. There is another thing that I want to highlight from this karyotype picture that we have seen here. You will notice that all chromosome pairs from 1 to 22 are very similar looking. And in fact, the chromosome pairs 1 to 22 are known as autosomes. They code for pretty much all characteristics that you have, with a few exceptions. But the chromosome pair 23 looks a bit odd here, doesn't it? Well, turns out that chromosome pair 23 is unique. They are called our sex chromosomes. This is because they contain genes that determine your sex, whether you are biologically a male or a female. Females have 2 chromosome X's, while males have 1 X and 1 Y. Well, this has been a lot of content to take in, hasn't it? Well, we are going to take a break in a second, but before we do that, there is one more concept that I want to talk about. And it is this slide that I wanted to introduce to you. This is a human life cycle. Remember, we start as this single cell that has both mother's egg cell and father's sperm cell fused together. This fusing together is what we call fertilization, and it results as zygote. Let's look at this by adding in what we have learned in this chapter so far. So both egg cell and sperm cell contained only half of all the genetic material of a normal human being. And we can also write this with these letters that we saw earlier on. N plus N is 2N. So to really make a new individual, we need to make sex cells that contain only half of the genetic material, and then we need to mix them to get unique combinations, and this is known as meiosis. And this is why making sex cells has this totally different process of cell division. And the rest, this process of mitosis, you are already familiar with.